Welcome to this video on the Barcode of Life Data System, or BOLD, a global cyber infrastructure designed for biodiversity informatics. We begin with Part 1, the Global Repository. This is the central hub where biological specimens are digitally linked to their genomic identities. BOLD serves as an informatics workbench supporting the acquisition, storage, analysis, and publication of DNA barcode records. It bridges traditional bioinformatics by assembling molecular, morphological, and distributional data. BOLD is a cloud-based platform for DNA barcoding. It acts as a Rosetta Stone, mapping unknown DNA sequences to Latin species names and enforces strict data validation. It was created and maintained by the Center for Biodiversity Genomics in Canada. BOLD has achieved a massive global scale, accumulating over 15 million sequence records. This unprecedented library manages a vast majority of curated data, representing hundreds of thousands of distinct species across the planet. In Part 2, we will explore the anatomy of a record. A BOLD record is a comprehensive, multi-layered digital profile of a biological organism, not just a string of DNA. A BOLD record is built upon three pillars: specimen data, which is contextual metadata, sequence data, the standardized nucleotide string and trace files, and specimen imagery, which includes high-resolution photographs. Specimen metadata is critical for traceability, providing geographic and physical context. Key fields include precise coordinates, identifiers, and the location of the physical voucher specimen. BOLD ensures transparency in sequence and trace files. It stores the assembled nucleotide sequence of the targeted marker gene, raw electropherograms, and exact details of the chemical primers used. Specimen imagery supports digital taxonomy. Photographs allow global taxonomists to visually audit the database and detect misidentifications, creating a virtual museum accessible online. Part 3 focuses on BOLD's architecture and algorithms, exploring the internal modules that power search, identification, and algorithmic species discovery. BOLD comprises 4 functional modules. The data portal allows open search. Management and analysis is a secure workbench for researchers. The identification engine is the core matching tool. Finally, the education portal is for students and educators. The identification engine takes an unknown FASTA sequence as input and aligns it against BOLD's curated library. The output is a diagnostic result, where high similarity to a single known cluster is considered a positive species ID. Barcode index numbers, or BINs, serve as a proxy for species. BOLD's algorithm automatically clusters sequences into operational taxonomic units, acting as a persistent species identifier before a formal Latin name is assigned. Analyzing bin concordance reveals relationships between named species in bin clusters. Outcomes include match, split suggesting cryptic species, merge indicating lumping, and mixture, which signifies taxonomic confusion. We now transition to Part 4, data integration and impact. Here we look at how BOLD connects with global initiatives and drives real-world scientific applications. BOLD is a foundational component of the biodiversity data ecosystem, actively collaborating with major databases. BOLD curates barcode data and pushes verified sequences to NCBI GenBank, which is the global public archive. BOLD also provides occurrence data to GBIF. Taxonomic representation in BOLD is heavily dominated by arthropods, making up around 70%. Plants, chordates, fungi, and others represent the remaining records. This reflects global campaigns focused on invertebrate populations. BOLD is experiencing exponential data growth. The intake of sequence records is accelerating dramatically, driven by advances in next-generation sequencing and the planetary biodiversity mission. BOLD has a significant global impact across several sectors, including modernizing taxonomy, supporting conservation biomonitoring in threatened habitats, and enhancing global biosecurity by identifying invasive species or illegal wildlife products.